Good morning, friends. So, here in this lecture, we will be discussing about the chromosomal aberration. And in that chromosomal aberration, there are two types of chromosomal aberration, that is structural aberration and numerical aberration. So, in this lecture, I will be discussing with you about structural chromosomal aberration. But let us start. What is chromosomal aberration? So, first of all, definition, you can see any abnormality of chromosome distribution, number, structure, or arrangement is known as chromosomal aberration. So, overall chromosomal aberration means any change of abnormality in distribution of chromosome, change in number of chromosome, structure of chromosome, or arrangement of chromosome. This is known as chromosomal aberration. So now we know the body, the structure or phenotype of an individual is controlled by genotype. That is, it is having genetic component, and genetic component along with environment control the development of an individual. So this genetic component along with environment will control the development of an individual. This, if there is any change in genetic component, it will lead to phenotypic variation, and that variation may be harmless or harmful or deleterious. Depending on that, this aberration, chromosomal aberration, has been classified. That is, it will give rise to the type of aberration. So, the changes and its severity or the type of changes has given rise to the different types of aberration. So, we'll see what are the different types of aberration. So, there are mainly two types of aberration: numerical chromosomal aberration and structural chromosomal aberration. So, here in this lecture, I will be focusing on the structural chromosomal aberration, which has been further classified into four types: the deletion, duplication, inversion, and translocation. So, these are the four main types of structural chromosomal aberration. And again, these types have been divided into different subtypes that we will be discussing in the later part of this lecture. So now, what are the structural aberration? So structural aberration are those aberrations that involve change in the chromosome structure. So as the name suggests, if there is change in chromosome structure, it will lead to structural aberration. The structural chromosome rearrangements are changes in the physical structure of chromosome that may result in birth defect, mental retardation, and increased risk of infertility. So, such structural rearrangement or change in the structure of chromosome, it may result in birth defect, mental retardation, or increased risk of fertility. So, these are the consequences of structural aberration. Now, when chromosome break and later rejoin in combination different from the original. So, how this structural arrangement aberration arise? So, first there will be break in the chromosome, and after that they will rejoin. They may rejoin, but they will rejoin in the different combination or in different orientation, and that will lead to the structural chromosomal aberration. And we have to remember that this structural chromosomal aberration is not a simple mutation. It is not mutation of genes, but it is the arrangement. It will cause the change in the order of arrangement of gene. It will not cause the mutation in gene, but it will result in the change in order or arrangement of genes. So again, a structural aberration. There are two type of structural aberration. The first one is the unbalanced structural changes. Unbalanced structural changes means there will be loss of some segment of chromosome, loss of some DNA. Here you can see there is break between B and C. This is the break between B and C, and you can see here this segment A and B is lost. A and B is lost. So the genetic segment is actually lost. Here again you can see there is break between the E and F and G and H, and now this F and G segment is lost. So in unbalanced structural changes there will be loss of DNA segment. So that is why it is called as unbalanced, because there is no balancing of genetic material. But there is balanced structural changes. In balanced structural changes you can see that there will be exchange of segment or transfer of segment from one chromosome to another chromosome. So in that you can see the non-reciprocal translocation. In non-reciprocal translocation you can see here the segment G, H and I is getting transferred to the non-homologous chromosome, another chromosome. Similarly, the reciprocal translocation, the mutual exchange, then paracentric inversion, pericentric inversion, we will see in the latter part of this lecture. What are these various structural changes? But as of now, you have to remember there are two types of structural changes: balanced structural changes and unbalanced structural changes, and few examples of balanced and unbalanced structural changes. So now you can see this structural changes. A structural aberration means alteration in chromosome structure, and there are two primary ways in which a structure of chromosome can be altered. The first one is the total amount of genetic information of chromosome can be changed. There will be change in the total amount of genetic information, and in that two aberration comes. The first one is duplication, another one is deletion. So what is duplication, what is deletion, we will be discussing in the latter part of this lecture. But as of now you can remember that this there will not be any change in the amount of genetic material. There will be change in the amount of genetic material in case of duplication and deletion, whereas there will not be any change of the genetic material in case of inversion and translocation. But there will be rearrangement of the genetic material. So let us see how this means. What about these four type of structural chromosomal aberration? The first one is deletion. So what is deletion? As the name suggests, it is the loss of segment of chromosome. This loss of gene present in the missing region. Loss of gene present in the missing region. There will be, if there is loss of segment of chromosome, the genes present on that segment will be lost, and it may occur either in both strand or in only one strand. Again, this deletion is of two types, that is terminal deletion and interstitial deletion. Here you can see in this picture the terminal deletion means there is cut between B and C, and you can see in the terminal deletion this A and B segment is lost. Whereas in case of this second part, you can see there is two breakage between B and C and D and E. And now this there will be two breakage and C and D segment will be lost. That means the segment will be lost from inside, not from terminal portion. So that is why it is called as interstitial deletion, whereas previous one was called as terminal deletion. So I hope this terminal deletion and interstitial deletion is clear to you. This is simply based on the name. That is interstitial means deletion from inside, and terminal means deletion from terminal side. So again you can see interstitial or intercalary deletion. It involves two breaks, we have already seen. There will be loss of segment and rejoining of the two chromosomal parts. So here again you can see by this picture the interstitial deletion. So there will be two break, and these break portion will be lost, and as a result new resultant chromosome will have lesser genetic material because some genetic material has been lost. So deletion was first discovered by C.B. Bridges in 1917. Now, the example of deletion is the cry-do-chat syndrome, or also known as cat-cry syndrome. It is mainly found in humans. It arises due to partial deletion of short arm of chromosome 5, and it will result in cleft palate, microcephaly, cat cry symptoms. The child will cry like a cat, and these are the symptoms we can observe in cry-do-chat syndrome. In cattle, it will lead to double muscled in blue Belgian cattle, and it will be caused due to defect in myostatin gene or deletion of myostatin gene. So these are the example of deletions. Now the second one is duplication. Duplication means presence of a chromosome fragment more than twice in the genome of diploid individual. So we know that the particular segment of chromosome will be present in two copies in a diploid organism, one coming from his mother and another from his father. But when there will be more than two copies of a particular segment, then that means there is duplication. So it usually arises by error in homologous recombination. So at the time of crossing over, we know there will be recombination where mutual exchange of segment of chromosomes will takes place between non-sister chromatid of homologous chromosome. But when there will not be mutual exchange, here you can see this yellow portion is doubled. So there will be two copies of this yellow portion in the same chromosome. So this is duplication. Duplication do not produce any drastic consequences like deletion. As we have seen in the deletion, there will be drastic symptoms, that is cat cry syndrome, or you have seen in the cattle, the double muscle. So this kind of symptom we will not observe in case of duplication, but there will be some symptoms I will see. This duplication in segment having centromere do not survive as to two centromere. Why? If there is duplication between at the point where there is centromere, that means the resultant chromosome will have two centromere, and that individual which is having two centromere will not survive, because these two centromere will go to the opposite pole or will have tendency to move towards opposite pole, and in that case individual will not survive. So this centromere, if centromere is doubled, individual will not survive. Example of this duplication is double bar I in Drosophila, which is resultant due to duplication of X chromosome. This is very common example you may see in case of duplication in Drosophila. And with the help of duplication, or with the phenomena of duplication, there will be formation of gene cluster or gene family. So as of now, you just remember the duplication will lead to the creation of gene family. What is gene family? This is not included in your syllabus. So just you remember, with the duplication there will be formation of gene cluster or gene family. So now there are different types of duplication. So here you can see this is the normal chromosome. This is the normal chromosome. Here you can see the tandem. First type is tandem duplication. As the name suggests, tandem means one after another. So here you can see the first segment portion is D and E. D and E portion is duplicated, and it is duplicated and joined after the first fragment. That is, D and E is duplicated and it is present there only side by side, one after another. That is why it is called as tandem duplication. Another one is reverse tandem. Reverse tandem means it is staying side by side but in the reverse direction. Here you can see D and E is changed to E and D. That means position is reversed. So that is why it is called as reverse tandem. And the third one is displaced on same arm. So it is duplicated. You can see D and E is duplicated and it is not staying there side by side, but it is displaced on the displaced on the same arm of the chromosome. Suppose this is P arm and this is Q arm. So originally it was present on P arm and now after displacement again it is present on the P arm, but it is not staying side by side. And the last one, that is fourth one, is the displaced on different arms. So it is duplicated, D and E is duplicated and it is translocated to different arm or displaced to different arm. So these are the four types of duplication: the tandem duplication, reverse tandem duplication, displaced on same arm duplication and displaced on different arm duplication. So now the third one is inversion. So what is inversion? It is a type of chromosomal aberration wherein segment of chromosome is reversed end to end. Here you can see this yellow portion, it is getting reversed. If you see the pattern, it is getting reversed. Break this, there is two breakage and after breakage inside segment will get reversed and again attached. So that is called as inversion. So this is rearrangement within the chromosome. There will be double break in the segment, already discussed. End to end rotation of the fragment between the fracture line and refusion of the fragments. So this is the this is how inversion is getting created. Now, what are the different types of inversion? So in inversion, you can see the inversion. The first one is paracentric inversion. Paracentric inversion. As the name suggests, para means little, or you can say away, little away. Paracentric, little away from the center. So what it means, this is the literal meaning. Now we can see the definition. The chromosomal inversion is inversion that does not involve the centromere. That means it is away from centromere. That is why the name is paracentric inversion. So what will happen here? Both break occur in one arm of chromosome. There will be break in one arm of chromosome, two break. Here you can see in this picture, there will be two break in one arm of the chromosome, and there will be end to end rotation of the fragment between the fracture line, and this again will refuse, and that will create the inversion. So this is mainly occurring at the time of segregation. At the time of segregation, here you can see in this picture how it is the pairing of chromosome, and at the time of segregation there is cut, and here you can see it is reverse end to end reversion, and then it will form the normal product and the inverted product. So this is about the paracentric inversion. On the similar line, you can see the pericentric inversion. As the name suggests, peri means around, centric means centromere. So the inversion takes place around centromere. So an inversion of segment of chromosome in which the centromere is included and break occur at both arm of chromosome. So you can understand this is the centromere. Here it is centromere, and break occur at both arm of centromere. Suppose this is Q arm and this is P arm. So there will be break in both the arm of chromosome, and that will lead to the paracentric uh, paracentric inversion. So here you can see this again, it will occur at the time of crossing over. At the time of segregation, you can see there will be the pericentric inversion. The inverted there, the sequence of gene will be inverted, and it will invert the centromere also. So this is about the inversion, pericentric inversion. Now you can see inversion is of two types. We have already seen inversion is of two types: paracentric inversion and pericentric inversion. And this is how this paracentric and pericentric inversion is created. So this was the summary about the paracentric and pericentric inversion. Now what are the difference between paracentric and pericentric inversion? The first of all, in terms of size and shape. So paracentric inversion will lead to the chromosome which is of same size and shape as of the normal chromosome. Means there will not be any change in the size and shape of the chromosome. Whereas in case of pericentric chromosome, it can change the relative length of the two arms, that is P and Q, depending on the how they are in case of pericentric inversion, we know the centromere is involved. So which portion, or you can say this whether this this may change the relative length of two arms of an acrocentric chromosome, therefore making it metacentric. So in case of acrocentric, if there is inversion, it may lead to metacentric chromosome. So if you you just think, you take a piece of paper and pen and try to draw in under what condition this acrocentric chromosome will be converted into metacentric chromosome, when there will be pericentric inversion. So again, that's how this what about cytological detection? How we can detect the paracentric and pericentric inversion? So for detecting paracentric inversion, you will have to go for banding technique, because their size and shape will remain same. So you cannot detect it by simple karyotyping. You have to go for chromosome banding. Whereas in case of pericentric inversion, there will be change in size and shape of the chromosome that can be detected simple by karyotyping. Then occurrence paracentric inversion occur very frequently, whereas pericentric inversion is less common. So this was about the inversion. Now this is the pictorial representation of inversion. You can see this paracentric inversion, it is away from centromere, so there is no involvement of centromere, and this segment is inverted and get attached, refused. Whereas in second case, you can see in paracentric inversion there will be involvement of centromere, and here there will be inversion, and this portion will get attached in reverse order. It will lead to paracentric inversion. So this was about the paracentric and pericentric inversion. Now the fourth one is translocation. So as the name suggests, trans means change and location means place. So there will be change in the place of segment of DNA or segment of chromosome. So a translocation involves the transfer of chromosomal segment to new location in the genome. So a particular segment will be translocated to new place. So it may be intrachromosomal or intrachromosomal. It may be within the chromosome or outside the chromosome. So here you can see the simple picture depicting the translocation. Now different types of translocations are there. So this translocation may be of three types. The first one is simple translocation, then reciprocal translocation, and Robertsonian translocation. The simple translocation is also known as non-reciprocal translocation or simply transposition. So it may involve 3 or 4 breaks. Simple translocation, one-way transfer of this is one-way transfer You have to remember this is one-way transfer of segment of one chromosome to non-homologous chromosome. So here you can see there will be breakage, and this break portion will get attached to the non-homologous chromosome. So this is leading to the simple translocation. Now, reciprocal translocation. Reciprocal translocation, as the name suggests, is the mutual exchange of segment between non-homologous chromosomes. So there will be simultaneous break in each of the two non-homologous chromosomes. There will be simultaneous breakage in the two non-homologous chromosomes, and then there will be exchange of genetic material. So here in this case, gene from one linkage group is getting transferred to another linkage group. So one linkage group, we know about the linkage group. So gene from one linkage group is getting transferred to another linkage group because there is involvement of two non-homologous chromosomes. But the amount of genetic material will remain same. So there will be only rearrangement. The segment of gene will get transferred from one chromosome to another non-homologous chromosome, but amount of genetic material will remain same. So this is the example of balanced chromosomal aberration. Now the reciprocal translocation is more frequent, most commonly seen in pigs, in animals, and examples are will be exchange of chromosomal segments between 1st and 16th chromosome, 4th and 14th chromosome, 6th and 14th chromosome, and so on and so forth. And it causes Down syndrome in humans. So you may be remembering, or you might have studied in your plus 2 classes about the Down syndrome. So there will be translocation between 14th and 21st chromosome. This will lead to Down syndrome. Another important resultant of this reciprocal translocation is the position effect. So what is position effect? It is because of the rearrangement of segment of chromosome, there will be change in the expression of genes, and this change in expression of gene due to different position of genes is called as position effect. So the rearrangement segment of chromosome may modify the expression of the existing gene in the region adjacent to the place of insertion. So such expression dependent on position is known as position effect. So this is an important term that is known as position effect. This is related to position dependent expression of gene. Now the third type of translocation is the Robertsonian translocation, which is actually it is centric fusion, and the variant of centric fusion is Robertsonian translocation. But this Robertsonian translocation is very popular. That is why the third one is known as or people are writing it as a Robertsonian translocation. Actually it is centric fusion. So what is centric fusion? So centromere of two non-homologous acrocentric chromosomes fuse to form the large metacentric chromosome. So what will happen? Two acrocentric chromosomes will come together and fuse to form the one large metacentric chromosome. But the thing is that there will be two centromere. The smaller arm of non-homologous which result from breakage are lost in the cell division. So the smaller portion, there will be breakage, fusion, and the smaller portion of acrocentric chromosome, the acrocentric chromosome, this is the position, this is the acrocentric chromosome, this is the smaller segment, this is the larger segment, again another one, this is the another the acrocentric chromosome. Now there will be fusion from centromere to centromere. So now the resultant chromosome will be like this. That is metacentric chromosome, large metacentric chromosome. But the smaller portion will also be there, like this and this smaller one. So this will be lost during cell division. there will not be any significance of this because it is not having any centromere. So this is about the centric fusion. Now again the Robertsonian translocation, or I already have discussed about the Robertsonian translocation. Actually it was given by Robertson in 1916 in insects. This is similar to centric fusion as I have told you, but the translocated chromosome has single centromere. So one centromere will be lost in due course of time, and that will lead to Robertsonian translocation. So one centromere in Robertsonian translocation and two fused centromere in the centric fusion. This is the difference between centric fusion and Robertsonian translocation. And the famous example of Robertsonian translocation is one translocation between first and 29th chromosome of cattle. It will lead to reduced fertility, early embryonic death, poor libido, poor quality semen, etc. So this is a very famous example of Robertsonian translocation of segment between first and 29 chromosome. So this first and 29th acrocentric chromosome will fuse together and will form one large metacentric chromosome, and the remaining portion will be lost. So this was about the Robertsonian translocation. Now there are few other structural aberration. In that, the first one is the ring chromosome. So what is ring chromosome? There will be deletion, terminal deletion of segment of a chromosome, and due to this terminal deletion on both the end there will be terminal deletion, and due to this deletion end of this chromosome will become sticky, and they will not join to any other chromosome, but they join together within itself. So it is the outcome of joining of two broken ends, that have resulted from two terminal deletion. So here you can see this is forming the ring chromosome. And the next one is the pulverization. So pulverized chromosome. So as the name suggests, pulverized means something like powder. So the chromosome with a pulverized appearance, so it will have pulverized appearance. It is characterized by fragments of stained material that were often clustered into discrete location on the spread. So here you can see this is the fragment of stained material, and they are placed somewhere else, not on the this is not a portion of any chromosome, but this is actually a chromosome which is pulverized. So DNA break and chromosome pulverized from error of mitosis. So during error of mitosis, this DNA will get pulverized. So this is the structural aberration, and this pulverization, ring chromosome, these are the mostly product of chemical mutation. Okay, this result may be due to the effect of chemical mutagen, and this is commonly seen in the people those who are affected from this Bhopal gas tragedy or the Chernobyl atomic leakage incidents. So in those individuals, in their karyotype, we can see the pulverized chromosome or ring chromosome. So this is all about the structural chromosomal aberration. I hope this whole structural chromosomal aberration is now clear to you, and the main thing is that everything in this structural chromosomal aberration is linked to the name itself. So there is no need of remembering spatially. You have to remember the classification, and if the name, you are remembering the name, you can easily draw the picture of that particular aberration. So in the next class we will be discussing about the numerical structural aberration. Till then, thank you. Thanks a lot.